Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
Have you ever really thought about how your body builds the very code of life, DNA, literally from scratch? Well, today, we're going to dive deep into that process. We're following the molecular journey of purine metabolism, from their simple beginnings to their absolutely critical roles in our health and sometimes in disease. It's a story of incredible molecular engineering, and it's happening inside you at this very moment.
All right, so here's our game plan. We'll start with the basic blueprint for purines, then we'll walk through exactly how they're built in what's called the de novo synthesis pathway. After that, we'll see how the final nucleotides are formed, how they're eventually broken down, and finally, we'll connect this whole process to the clinical reality of a condition called gout. Okay, let's jump in. I want you to think of your body like this massive, incredibly complex construction project. And before you can build anything, what do you need? A blueprint, right? Well, in our cells, purines are a huge part of that blueprint. They are the A's and G's in your DNA and RNA, the very letters that write out your entire genetic story. And as if that weren't enough, they also form ATP, the molecule that's basically the universal power source for everything your cells do. So what's the whole point of all this? The end goal is to build something called purine nucleotides. And while this happens in a lot of places, the liver is kind of the main factory here. Now, our bodies are super efficient and can recycle purines, but for right now, we're going to focus on what's called the de novo pathway. De novo is just Latin for from the beginning. And that's exactly what we're doing, building these complex molecules right from the ground up. And this is what I find so amazing. Look at this intricate two-ring structure. It doesn't just pop into existence, it's assembled piece by painstaking piece, atom by atom. And the building blocks? They're actually super common things. Amino acids you get from your food, and even the carbon dioxide you breathe out. It's just a beautiful example of how incredibly efficient our cellular machinery really is. Okay, so let's get into the nitty-gritty of how this construction project actually works. The de novo synthesis pathway. Think of it as a biochemical assembly line. It's super precise, it requires a lot of energy, and it needs a whole crew of specialized enzymes to get the job done right.
Right. Every good building needs a solid foundation. In this case, that foundation is actually a sugar molecule. An enzyme zips in, gives the sugar a jolt of energy, and turns it into a molecule called PRPP. You can just think of PRPP as the prepared slab of concrete we're about to build our purine on. It's the starting point.
And the build itself, it's a 10-step process. It's like this perfectly choreographed molecular dance. It all kicks off with one specific addition, and this first step is what we call the committed step. Once it happens, there's no going back. The cell is committed to building a purine. This step also sets the pace for the entire operation. From there, it's a series of additions and tweaks, all culminating in the very first complete purine, a molecule called IMP.
Let's take a closer look at the first half of this, steps 1 through 5. You can see our PRPP foundation right there, and piece by piece, new molecules are added in a very specific order. Then, bam! Step 5, the first ring snaps shut. Now this part of the process costs energy in the form of ATP, which really shows you how important and valuable this final product is to the cell.
All right, now for the home stretch, the second half of the build. With that first ring done, the cell gets to work on the second one. A carboxyl group is added, then an amino acid, aspartate. A piece of that gets clipped off, leaving behind a nitrogen atom. A final formal group is added, and then, in step 10, the final ring closure happens. And what do we have? We've got IMP, the parent purine nucleotide. This is the molecule that everything else is going to be built from.
So we've built our base model, our platform molecule, IMP. But to build DNA and RNA, the cell needs two very specific purines, adenine and guanine. So the next job is to customize IMP into these final, ready-to-use forms. So our IMP molecule comes to a fork in the road. It can either become AMP or GMP, the two purines we need. And what's really genius here is how the cell balances the production of both. Check this out. To make AMP, it uses energy from GTP. But to make GMP, it uses energy from ATP. This kind of cross-regulation is so clever. It makes sure the cell never has too much of one and not enough of the other. It keeps everything in perfect balance.
You know, building these things from scratch is a ton of work, and our cells are all about efficiency. So of course there's a shortcut. It's called the salvage pathway. Instead of building from the ground up every single time, this pathway just recycles purines, whether from our diet or from old cells breaking down.
It's way more energy efficient. And for some cells, like our red blood cells, which can't do the whole de novo thing, this is literally their only option.
Okay, so we've seen how purines get built and how they get recycled, but what happens when they're old and not needed anymore? Well, the body has to have a way to break them down and get rid of them, and that brings us to purine degradation. So when it's cleanup time, both AMP and GMP are taken down a very similar disassembly line. They get stripped down piece by piece until they both end up as this intermediate molecule called xanthine. And from there, one really important enzyme, xanthine oxidase, steps in and converts xanthine into the final waste product, uric acid. This uric acid then gets shipped off to the kidneys to be flushed out. This whole system, from creation to destruction, is a really delicate balancing act. So what happens when that balance gets thrown off? Well, the consequences can be pretty severe, and they can lead to an incredibly painful condition you've probably heard of, gout.
The root cause of gout is a condition called hyperuricemia, and the name tells you everything you need to know, right? Hyper means high, uric is for uric acid, and emia just means in the blood. So it's simply a state where you have way too much uric acid floating around in your bloodstream.
So what's a normal level? Well, for males, a healthy concentration of uric acid in the blood is anything below 7 milligrams per deciliter. At that level, it stays dissolved no problem, and the kidneys can handle it just fine.
But what happens when that level creeps up above the 7 milligrams per deciliter mark? Well, the blood basically becomes saturated. It can't hold any more dissolved uric acid, and so it starts to precipitate out, forming tiny razor-sharp crystals. And I think you can imagine where this is going.
And this is where all that abstract biochemistry becomes very, very real and very painful. Those sharp little uric acid crystals don't just hang out, they start to build up in our joints, especially in places that are a bit cooler, like the big toe. This buildup triggers a massive, aggressive inflammatory response from the body, and the result? The excruciating, debilitating pain of a gout attack.
Now, while a lot of things can contribute to this, about 10% of cases come from the body just flat out overproducing uric acid, often because of genetics. For example, you could have an overactive PRPP synthetase, remember that enzyme from the very first step, that just puts the whole assembly line into overdrive. Or you could have a defect in that recycling enzyme, HGPRTase. If the salvage pathway is broken, the body ends up making and breaking down way more purines than it needs to. Either way, the end result is the same, way too much uric acid.
Yeah, for anyone who's actually experienced a gout attack, it's something you never forget. It often hits you out of nowhere, usually in the middle of the night. Just this blinding pain, redness, and swelling in a single joint. And while the big toe is the classic spot for a first attack, really any joint can be a target.
So, that brings us to a final thought. We've gone on this incredible journey, from simple atoms all the way to a complex metabolic pathway. And then we've seen how a disruption in that pathway can lead to a really painful human disease. This kind of deep biochemical understanding, it's not just for textbooks, it's the absolute bedrock of modern medicine. And so the real question becomes, how can we take this incredibly detailed knowledge and use it to design therapies that can precisely target these enzymes? How can we better manage, or maybe, one day, even prevent these kinds of metabolic disorders?